remember, messenger RNA is the copy of the gene. It will leave that nucleus through these little openings called nuclear pores. And then will travel to the ribosome where the protein will be made. Yeah.
That reminds me, the 3 RNAs that we need to remember are messenger RNA, transport RNA.
Transfer.
Oh, transfer RNA, thank you.
rRNA, which is the ribosome, which is what we're talking about here.
Thank you.
Ribosomes is going to be partly genetic material, RNA, and partly protein. What I want you to remember, there are 2 kinds of ribosomes. Ribosomes that are just floating around in the liquid inside the cell are called free. They're not attached to anything. They will make proteins, and that's what ribosomes do, make proteins. They will make proteins that are then used by that cell. In other words, it may be to repair an organelle, or it could be to repair the membrane. But the proteins they make will be used by the cell. In contrast, there is another organelle called the endoplasmic reticulum, abbreviated ER. If the ribosomes are attached to the ER, those ribosomes will make proteins for the purpose of taking it outside of the cell, such as insulin is made in the pancreatic cells. Insulin has to leave the cell, get into the bloodstream for it to do its job. So it is made on the ER. The reason that it is made on the ER is the ER is going to be what helps to take that protein out of the cell. It's going to be the way that it comes out. And it does that by packaging it in kind of a little globular membrane that will let it just fuse through the fatty layers of the cell. So ER has ribosomes that make proteins that leave the cell. Free ribosomes stay in the cell, the product, protein. These shows the units of the ribosome. We'll go into those in detail much later. So don't spend too much time thinking about those. Yeah.
So the endoplasmic reticulum has so the ribosomes make the proteins there, and then the proteins are packaged and they can leave the ER and leave the cell. Okay, so that's where basically the main point is that the ribosomes attached to the ER make the proteins there. So, okay.
Endomembrane system is another system that's important. This is the way cells communicate. These are the parts. These are the membranes within the cell. Remember we said prokaryotes don't have these membranes, eukaryotes do. These are the parts that make it up. The nuclear envelope contains genetic material. Endoplasmic reticulum packages proteins to be moved. The Golgi apparatus helps the ER basically. They both have different jobs, but for now just think of both the ER and the Golgi do these. You'll find later when we study it, the ER makes little membranes, the Golgi uses that to package it, yeah.
So would you like us to memorize the components of all of them?
Yeah. Nuclear envelope, DNA, genetic material, ER, and the Golgi apparatus package and move proteins. Lysosomes have to have a membrane because they have very nasty enzymes in it that breaks down cellular parts and debris. They're the garbage dump, so they need to be separated from the rest of the cell. Vacuoles in a plant can open and close to allow water to come in. Plasma membrane is the one that protects the internal environment of the cell, simply enough. Plasma membrane protects, controls the internal environment of the cell. Many of these membranes can be connected to each other or other little parts that connect them, as it says, the vesicles. This is what I've just told you about now, okay? So you're not getting new material here. Endoplasmic reticulum says smooth and rough. Rough ER is the one that has the ribosomes. Now, smooth ER that does not have ribosomes is obviously not involved in the role of packaging protein. It has other jobs. It is important to know what they do. That's a rough ER. This blue part is the ER, and the red is the ribosomes. I do want you to know exactly what the smooth ER does. Even though it's not involved in the packaging of proteins, it plays a vital role. Makes the fats that the cells and your body needs. Makes them. It'll take the basic atoms, elements, and put them together to make the lipids that your body needs, like the phospholipid membrane, for example. It can also metabolize carbohydrates. And the third one right there is enormous. It detoxifies drugs, poisons, anything that needs to be broken down and removed out of your body that's chemical. You will find some people may be on a type of high blood pressure medicine that may work for 5 years and then stop. The reason is, after a while, your body starts to figure out, we're getting this constant influx of this drug and we're not breaking it down fast enough. So it makes more smooth ER. Suddenly, almost as soon as the stuff hits your system, it breaks it down so the drug doesn't have enough time to work. Detoxification occurs in the liver cells. So liver cells will have a very high amount of smooth ER because that's the organ's job, is to break down. If you take something like penicillin, it usually takes 8 hours in order to get 8 to 24 hours to get what is called the blood level, that effective level where it works. And it will usually take, like if you were taking penicillin, it'd take about 48 hours for it to totally leave your body. And you would still have the effects. That is being broken down by the smooth endoplasmic reticulum. Alcohol, drugs, legal or illegal, whatever. One of the bad things about some illegal drugs like cocaine, if you take alcohol and drink it, it damages the liver cells, but they will recover. The liver is very hard and tough organism. It'll get better if you give it a chance. If you take some of the drugs like cocaine, it don't damage them, it kills the cells. The cells can't repair if they're dead. So there are some drugs when you take, the cells just die, they're done. One of the worst drugs that you can take right now for your liver is Tylenol. Acetaminophen destroys liver cells. And there is recommendations now, if you're a patient with arthritis, do not take Tylenol for the problem. Take ibuprofen or aspirin. Don't take Tylenol. The long-term use of Tylenol is not good. One of the most damaging things that you can do to your liver is to take Tylenol and alcohol together. It magnifies the destructive power many, many times. Many of the big drugs, oxycodone, hydrocodone, they have other things mixed with them, and one of the more common is acetaminophen. So when you start mixing those drugs together, you're going to destroy the liver. It has been found that one single dose of some of these in some people can make you addicted. Take it one time, and your body shuts down the production of the other things it needs. It can do you a lot of damage. One time can kill you. So the liver, the smoothie ER got a big job to do. And if you stress it, it will start to make more and more smooth endoplasmic reticulums. Now your blood pressure medicine don't work. The doctor's got to switch it to a version that your liver's not designed to fight now. And it makes calcium, or stores calcium. So remember those four things from the smooth ER. The rough ER, I don't really want you to remember anything yet, except the rough ER has ribosomes that make protein. Then that protein is going to be modified by the ER and packaged with the Golgi and moved out of the body. The purpose of the Golgi is like a little blob of fat, and when you make the protein and it gets ready, a blob of that fat comes around the protein, it migrates through the cell, it gets to the fatty layer of the membrane, and it can just kind of fuse through it like a ghost walking through a wall. It just fade right through, and that's the way things get out into the blood. Notice it says it modifies products of the ER. It's talking about the protein and the membranes and so forth. It can make some molecules, but the big thing for the Golgi is transport vesicles. That's those little blobs I said that just form. Remember, the lysosome is basically the garbage dump of the cell. When you have parts of the cell that break down, go bad, they have to be removed and replaced, you have to get rid of the junk that's left over. Lysosomes contain enzymes that will break down these no longer needed parts into their basic components, and they will leave via usually the urinary system. Notice lysosome enzymes work best in an acidic environment. You obviously don't want the rest of the cell to be exposed to an acidic environment. So we have a membrane around it, which makes it part of that endomembrane system, where it can do its job, its function, isolated so it doesn't hurt anything else. Some cells can eat each other. Basically, white blood cells, for example, phagocytosis. That's a way to get rid of a bad cell like a bacteria that is not good, or if you've got a cell that is died, it's not doing its job anymore, another cell like the white comes up, just engulfs it. We say eat it. It basically just wraps around it and then uses these lysosomes and things to digest it, to get rid of it. A vacuole is basically just like a door to a cell. Plants have these a lot. Things that live in water, some protozoans might have something similar to that where they can let water in and out. Mitochondria, I want you to remember, they release energy. When we eat food, that food is released by way of the mitochondria. That is where cellular respiration takes place. Mitochondria, as it says, generates, does not create, but generates ATP. There's a law of thermodynamics that states we cannot create nor destroy energy. Only thing we can do is change its shape or form. Chloroplasts contain chlorophyll, and that's where photosynthesis occurs, and that generates ATP by way of sunlight. Peroxisomes are little organelles that do some of the tough work, same thing almost like lysosomes, but they are basically going to break down hydrogen peroxide before it gets in your system and destroys it. If your body goes through chemical reactions and creates hydrogen peroxide or peroxide type compound, that will be broken down within the peroxisomes. Okay. process, they are oxidative. You don't know what an oxidation reduction reaction is yet, you will. Evolution of mitochondria. Let me just put this down and save you a lot of trouble. There's a theory that one time in our far past history, evolutionary history, our cells absorbed bacterial cells into them. And the reason was the bacterial cells were so efficient at getting energy, much more efficient than our cells. So our cells just engulfed some of that bacteria and started making.
Have your attention, please. May I have your attention, please? The following is a test of the Kansas Severe Weather Warning System.
Okay.
This is only a test.
The bacteria went into the cells, and then we used them to make energy. So that is called the endosymbiont theory that you see. That's the theory that our cells incorporated bacterial cells in order to produce energy. That's all you need to remember about it. This shows how that could have occurred. We got bacteria out here, our body just takes it in, and our mitochondria are thought to be remnants of that bacteria. We're not going to spend a lot of time talking about the mitochondria, but remember inside of it, it does have a fluid, a space its own called the mitochondrial matrix. And if you look back, you can see it's got all these little folds in it. That is to increase surface area so it can release a lot of energy.
Did you say that included a fluid?
Yeah. The mitochondrial matrix is the opening and the fluid that's inside the mitochondria.
Oh, so the matrix is the fluid that's inside. Okay, thank you.
Basically, yeah. Chloroplast captures light. We're going to cover that in good detail when we talk about photosynthesis. So I prefer to cover chloroplast when we're talking about photosynthesis and mitochondria when we talk about cellular respiration because they are related. This does, I do want you to understand a little bit about the structure of a chloroplast here. Thylakoids are sacs. Those sacs, like a coin, are stacked on top of each other. They make the granite. The stroma is the fluid inside of those. We will see that later when we talk, like I say, about photosynthesis, so don't spend a lot of time on it now. These are the peroxisomes, and notice it says these are specialized little compartments for projection. They produce hydrogen peroxide and convert it to water. Now, what that means is there's some things when we break them down in the body, the resultant chemical that made is hydrogen peroxide. You can't leave hydrogen peroxide out in your body if you've ever had it cut. You see now it bubbles and oxidizes what it does to tissue. So you don't want that inside of the cell. So the peroxisomes, instead of doing this in a lysosome where it may get out, we've got a special membrane. So that will then break down those chemicals. Peroxide will be made, and then all it has to do to turn it to water is what? Get rid of one oxygen. We've got water. That's all you need to remember about peroxisomes. They break down the product of hydrogen peroxide into water. Cytoskeletons, I want you to read this. 3 basic kinds are microtubules, microfilaments, and intermediate filaments. These do 2 things primarily. Number 1, they give structure or make a skeleton for the cell. They form a network, as you can see right there. Those colored things are the microtubules and filaments. So they give structure so a cell keeps its shape. And they also, things can move along those filaments almost like a rail car attached to a big cable or railroad tracks. They can move things, so some molecules can actually move them. So their function is for structure and transport. You really don't need right now to remember much about that. Don't worry about these proteins and the filaments. We'll talk about those later, because they're more important.
So you said we don't need to know about the motor proteins?
Now, just know those three basic kinds of cytoskeleton, like the fibers and so forth. We will talk about centrioles and centrosomes when we talk about DNA. If we talk about it now, you won't probably get it. So all of this is talking about the microfilaments and all of that. Don't worry about it. So when we get from basically know that page, and then just skip over to cell walls of plants. And it's about time, so we'll stop there. We've only got 5 slides left. We'll finish that off on Friday, and then do the review.
Did you say we just need the page about microtubules?
Just go to that one page